Hello, this is Andrés Martinez from Argentina, and together with Mónica, Andrea, and Maité, would like to share some of our results on the preference and performance of Drosophila Suzukii in different berries in Patagonia. The spotted wing Drosophila is a highly polyphagous fruit pest native to Asia, which in the last decade has widely expanded its range and become a serious pest in America and Europe. Adult females oviposit on soft-skinned fruit where larvae develop, causing immediate damage that results in fruit rotting. The life cycle can be as short as 15 days in places where climatic conditions are adequate, resulting in high population levels within short periods of time, with economic losses reaching up to 90%. Fruit variety could be important in determining female oviposition preferences and larval development. Factors such as fruit sugar content, color, and skin hardness could be important in determining female choice and larval development. In this context, our aim was to analyze the fly's phenology and compare, through field and lab trials, the fly's preference and performance between raspberry and blackberry cultivars in northern Patagonia. We hypothesized that since sugar is one of the main resources needed for development, its concentration will impact in female fruit choice and larval development. The study was conducted during the austral spring and summer of 2019 and 2020 in the Patagonian Andean valleys of Argentina, where berries have been cultivated for more than 60 years, with approximately 300 hectares of farms with an ideal climate for this activity. We surveyed 2 farms with 2 raspberry cultivars, Himbotop and Autumn Bliss, and 1 blackberry cultivar. In order to quantify adults in flight, we used vinegar-baited traps as shown here. 18 traps were set in total, 3 per cultivar per farm, between November and March, with revisions carried out every 15 days. We also surveyed adult emergence from 20 grams of berries collected at each trapping location during February, which is the main co-occurring fruiting period of the cultivar surveyed. Naturally infected berries were reared under controlled conditions in the lab and emerging adults quantified. Since data varied between dates and farms, we calculated 2 indexes, a female flight index that took into account the number of females captured in vinegar traps in a particular cultivar, date and farm, divided by the mean captures of that date in that particular farm. This index was used as an indicator of possible biases in female flight between cultivars. We also calculated a second index with the flies that emerged from fruits using the same logic. This one was used to compare oviposition and larval development rates between berry cultivars. So, results show that captures in vinegar-baited traps are relatively low until the end of summer, moment when flies population peaks. Unfortunately, we could not obtain data for the complete flight season because of COVID restrictions in mobility. We also observed no significant differences between raspberry cultivars in female flight index and the fruit emergence index. With raspberry data pulled together, we observed no differences in female flight rates, but we did observe differences in the fruit fly emergence index, with significantly more flies emerging from raspberries than from blackberries. Interestingly, we did not observe statistical differences in the sugar concentration between berry cultivars. A more detailed correlation analysis shows no indication of higher fly presence in berries with higher sugar concentration. In fact, the fruit emergence index seems to be negatively correlated to sugar concentration in blackberries. So finally, we can conclude that the highest population levels are observed at the end of the summer. We also did not detect behavioral differences in flying females or emerging adults towards raspberry cultivars. Additionally, we observed no biases towards raspberry or blackberry on flying females. But we did detect higher emergence of flies in raspberries than blackberries. This could be due to females ovipositing more on raspberries, in addition to higher development rates on this fruit. Interestingly, sugar does not seem to impact as expected on preferences and performance for any of the berries tested. Further studies are needed to understand the drivers behind the observed differences in emergence rates. For instance, berry chemical and visual information could be affecting oviposition and development rates, in addition to particular yeasts within the fruits that could affect larval nutrition. Finally, we would like to thank the financing bodies that allowed this work to be carried out. That's all for now. Thank you very much.